Okay, so today is our 19th lecture. So we finished the system biology module, and we now going to talk about something else. So this will be the first of the two classes on a module on 3D structure. So today we're going to talk about the 3D structure of the DNA of the chromosome. And on Wednesday, we're going to talk about the 3D structure of the RNA and the protein. Okay, so today's agenda, we want to think about DNA not as a element that code the gene sequence, but we want to think about it in terms of 3D molecules, because DNA and chromosome are also in 3 dimensions. So their structure also have function. And there are ways that we can study the function, and there are also computational models that we can use to understand the structure of DNA. So we probably know from basic biology that during cell division, we need to condense DNA. We need to make sure that the 3 billion nucleotides that we have in our genome is condensed into the signature X shape, that became very small, so that we can duplicate the DNA and then divide it in between daughter cells. So people knew for a long time that the structure of the DNA has to be organized in some way. And we learned from the epigenetic class that there are proteins, histones, that together form a nucleosome. And nucleosome can bind to DNA and package the DNA so that you can coil the long strain of DNA down into a regular structure. But even more than that, people know that when you have active and inactive DNA, like heterochromatin and euchromatins, they are not just chemically labeled like methylation, but they also, in terms of structure, they also localize in a different location in the nucleus. So the heterochromatin or inactive chromatin are usually recruited to the nuclear lamina, so away from the transcription factor, away from the transcription machinery. So when DNA needs to be activated or deactivated, it's not only just about labeling their status, but also moving them to a specific location, specific compartment inside the nucleus. So people also found this association for a long time. And we also learned that in some disease, it's caused by DNA duplication. So it makes sense that if you alter the DNA, you copy the gene, you can cause some disease. But you might be surprised that just getting an additional copy of the gene might not be sufficient to cause the disease, because when we get more copies of the gene, we have high expression level, but it's still within the level that the cell can tolerate. But some DNA duplication that resulted in the disease is because on top of DNA duplication, the part of the DNA that duplicates, as we will learn in a later part of this class, is that this new DNA also forms its own independent 3D structure. And with this independent 3D structure, it changes the regulation of the gene. So it's not just about the fact that there's an additional copies of the gene, but also that the additional copies now exist on the DNA, on the chromosome, in the region that are regulatorily different than the original one. So people found that the organization of the DNA also contribute to the regulation of gene expression and also the cause of disease. So just to give you a preview of the data that we are going to see a lot of today, today we're going to see a lot of heat map like this. So this heat map, the X and the Y axis will be coordinates on the genome. And the color inside, if it's red, that means that these two parts of the DNA, maybe on different chromosome, maybe on different region of the chromosome, they are nearby in 3 dimensions. So even though they are far apart on the genome sequence, in 3 dimensions, they are folded next to each other. So you can see patterns of interaction between different regions of the genome. And people found that during cell development, when cells change from one state to the other state, they need to activate some gene. And enhancer needs to fold onto the transcription start site in order to facilitate the activation of the gene. So there is some movement of the DNA in terms of the structure. And we can identify these changes if we use molecular assay that target the 3D structure of the DNA. We will see a lot of visualization like this in today's class. And during cell division, we can see that the chromosome kind of condense, and then after the cell division, the structure now is more less dense and return to the normal state. So this dynamics of condensation of the DNA, relaxation, condensation, can occurs every time we have cell division. So the DNA is really dynamic. Every cell division, there is a large scale movement on the DNA. And in later slides, we will learn about the proteins and mechanisms that can explain this phenomenon. Because if the cells doesn't have a way to regulate the 3D structure of the chromosome, you cannot organize the cell division, you cannot organize the cell development. So there are proteins and other mechanisms that help cells facilitate this process. And people found that the structure of the DNA also contribute to the mutation, the location where we found mutation. So if we think of mutation as like an error in DNA repair machinery or DNA damage from radiation, from UV, because DNA are also in a three-dimensional kind of molecules, the location where the mutation occurs also localized at certain location on the DNA, not on the genome sequence, but on the 3D structure of the DNA. So people found that there are some specific locations in the genome that are frequently mutated in cancer. And the interesting thing is these locations, if you go read the paper, they will say that these locations are actually clustered in 3 dimensions. It's not like it's randomly distributed throughout the nucleus. It's focusing on certain area on the DNA that are localized in certain part in the nucleus. And you have a lot of cancer accumulating in that area. And not just in cancer, when people study variation in DNA from one plant to the next in the Arabidopsis, they also found that there are locations that are frequently mutated across the variant of the plants. And these locations also tend to cluster in the same 3D space, not on the DNA sequence, but in the 3D space. And people also found that there are certain elements on the genome that if you mutate, it will cause a lot of disruption to the chromosome 3D structure. So we will found in later slide that these CBS, like CTCF, which is the name of the protein, the binding site of CTCF on the chromosome, CTS is a protein that can bind to the chromosome. This location, if there's a mutation, it disrupts the binding, the binding motif now change. It can cause a large-scale disruption in the chromosome 3D structure. And these locations are frequently found in cancer because when you mutate these areas, you can change the regulation of genes that are near each area. And so people found that this understanding the 3D structure of the chromosome, like understanding how the 3D structure is formed, also help to understand disruption in disease as well. So people have found that in general, we can think of the 3D structure of the chromosomes in several scales. On the largest scale, if you fluorescent label individual chromosome, like chromosome 1, 2, 3, 4, 5, with different colors, the first thing you will find through microscope is that the chromosome, each chromosome, will fold into like a ball separately. So let's say chromosome 1 is in red, or chromosome 3 is in red. So chromosome 3 will be folded like as a globule, as a ball, and placed at a certain position inside the nucleus. It's not like chromosome 3 is free, flowing throughout the entire nucleus. We found that the chromosomes fall into a tight globule, and it's occupy a certain location in the nucleus. And people found that chromosomes doesn't entangle with each other. Basically, they don't fold together, like they fold separately. And that's good news, right? Because if you have multiple chromosomes, like we have 23 chromosomes, right? If the chromosomes kind of entangle with each other, imagine that during cell division, the DNA has to untie themselves like from each other. And actually, if you read kind of paper written by physicists, they will explain that if you model DNA as a polymer, like a string, very long string, and if you allow the string to be entangled, like forming a knot inside your nucleus, the time scale it takes for the knot to untie, like if you want to untie, if you put like a lot of cable in your pocket, right, they will all clump up and then they will entangle with each other. The time scale that you need to untie them is actually longer than the time scale of cell division. So with kind of basic principle from physics, they can argue that in biology, if there is no other forces that organize the DNA, there's no way that we can have a proper cell division. Everything will be tied up together. And if you zoom in further on the chromosome, on individual chromosomes, you will find that each part of the chromosomes also fold into separate 3D domains. Like how when you imagine protein structure, like we have different domains. One for binding to DNA, one for catalyzing some reaction. We found that the same phenomena occur on DNA. Different parts of the chromosome form these domains, which people term topologically associating domains. So it's kind of nearby DNA that fold together. And people found that there are many functions for this, which we will explore in later slides. And on the smaller scale, like if you zoom in on a very small section of the DNA, there are usually loops. So loops mean enhancer, for example, when it reach the transcription start site, the DNA has to form a loop so that these two loci can be next to each other in 3 dimensions. So these activities, from looping to topologically associating domain formation to chromosome kind of territory formation, these are organized inside the nucleus. And in early study, they look at the location of the chromosome of the specific element on the chromosome. Hopefully you remember centromere, telomere. So centromere is the central part of the chromosome. Each chromosome has one centromere. So people know from back then that the centromere from multiple chromosomes aggregate with each other. So these are the domains that can interact with each other. The telomere as well, the telomere from different chromosomes can also aggregate with each other because of the sequence that are similar on them. So people found that if you visualize, if you fluorescent kind of label the centromere, the telomere, and also the position for the ribosomal DNA in different cells, you imagine this in many cells, and you align the image together. You put the centromere, you rotate the cells until the centromere are together at the same single location. People found that fixing the location of centromere also show the position of the ribosomal RNA, which will be on the opposite side of the centromere. And the telomere will be on the nuclear membrane, on the surface of the nucleus, and away from the centromere. So people found that there are some basic organization inside the cell. Once you fix the location of centromere somewhere as a frame of reference, you can tell that the ribosomal DNA, whichever chromosome is on, will be located opposite from that side. And the telomere will also be flexible, but it will be on the surface of the nucleus. And even more interesting is if you focus on a certain location of a gene, like GAL1, GAL2, like in yeast. So these genes are response to the nutrient, to the use of glucose or galactose in the nutrient. So when we think of gene activity, like in terms of molecular biology, we tend to think of, gene being activated or deactivated, like transcription factor might bind to the promoter and then activate the transcription. But that doesn't take into account that genes, the DNA sequence of the gene, also float around inside the nucleus. So people found that the location inside the nucleus also has some effect on the activation of the gene. For example, GAL1, if the nutrient is rich in glucose, so everything that we are seeing here, we are fixing the location of the centromere. So we have a frame of reference, which is the location of centromere. So we found that the GAL1 position is near the centromere and very stable. So if you assume that like the location of GAL1 doesn't really matter, so inside the nucleus, GAL1 should be able to be anywhere, depending on the cells. In some cells, GAL1 might be on the top. Some cells, GAL1 might be on the bottom. Some cells, GAL1 might be on the left. Some cells, GAL1 might be on the right. But they found that if we fix the location of centromere as a frame of reference, the location of GAL1 is pretty deterministic. It's also localized near the centromere, near the same coordinate of the centromere, in glucose condition. But if we change from glucose to galactose, we found that the position of GAL1 is now not unregulated. It can be anywhere inside this half of the nucleus. So under galactose condition, GAL1 is no longer controlled. It can flow to anywhere. If we look at GAL2 in glucose condition is located in the middle between centromere and the ribosomal DNA. Under galactose condition, GAL2 is still in the same region, but now it's kind of spread out a little bit. So we can see the dynamics when genes need to be activated or deactivated. It's not only just about transcription factor binding, not only just about changing the state of the chromosome, it's also about positioning of the chromosome inside the nucleus. So there may be some region in the nucleus that are rich in polymerase or transcription factor. If the cells can move DNA of a gene into that region, it can facilitate the transcription. It brings the DNA to where there is a high concentration of polymerase of transcription factor. So when people think about 3D structure of the chromosome nowadays, we think of it as a regulation mechanism. If you can bring multiple genes that share the same enhancer together, that means that a transcription factor might bind to the enhancer, and then that enhancer can activate multiple genes, because enhancer can move to gene 1, activate the transcription factor and the enhancer can then move to a second gene, also activate the transcription, and then move to the third gene and activate the transcription. So people found that by having some organization, some 3D structure on the DNA, allow the cells to organize gene as a group that should be activated or deactivated together. And this is just one of many mechanisms that 3D structure of the chromosome help the operation inside the cell. We'll see more of this later. So let's talk about now the assays that we can use to study the structure of the chromosome. In the epigenetics lecture, we learned about the assay that we can study the state of the chromosome, whether it's methylated or unmethylated, whether it's open or closed, whether it's accessible or not, whether it's bound to the nucleus. Now we're going to talk about the assays that help us understand the 3D organization. So, for example, there are a series of experiments called chromatin conformation capture. As the name suggests, it's chromatin, it's conformation of chromatin, and we want to capture it. So what happens is when two different regions on the chromosome, if they fold together in 3D space, when any molecules come close in 3D space, as a chemist, they can induce a cross-link, a covalent bond between the two interacting molecules that are nearby. And once we introduce a cross-link, we can pull down the the cross-linked DNA, usually with biotin. And then once we use restriction enzyme to cut the DNA, and then we can ligate it. So if we follow the diagram, we have two pieces of DNA, maybe from different chromosomes, maybe from different regions on the same chromosome. They are linked together because they are near each other in 3 dimensions. We pull them down, we cut with the restriction enzyme, and we ligate it, one end of it. So we create this kind of a hybrid DNA fragment, where one end comes from one region of the genome, the other end comes from a different region of the genome. And then we can sequence this hybrid fragment. And the pair-end sequencing will tell us that which part of the genome the orange segment is from, which part of the genome the blue segment is from. And using this knowledge, we can say that the blue segment may be from chromosome 3, and the orange segment may be from chromosome 5. These two segments are near each other in 3 dimensions. So if we have a lot of sequencing data of these hybrid fragments, we have information about which position on the genome are near each other in 3 dimensions. So chromatin conformation capture actually consists of a class of techniques from 3C, 4C, 5C, high C. So the increasing number here generally means increasing throughput. So in the past, with 3C technique, you have to specify which location on the DNA to study, and you have probe, PCR probes that are specific to that region. So you can only study interaction between 2 specific loci at a time. So you have to know where to look. But that's not good, right? If you want to study DNA organization and you don't know anything beforehand, you cannot choose your PCR target. So people invent 4C and 5C and high C so that nowadays you don't need to know anything. You just induce cross-link between DNA from any location on the genomes that are near each other in 3 dimensions. And we do high throughput sequencing, and we got the interaction between every location on the genome. But as a biologist, you might want to know about these separate techniques because your biological question might involve specific locus and not the entire genome. If you can perform a targeted experiment, that saves you the resource and the cost. Okay, but anyway, so once we perform chromatin conformation capture, we are going to get data that tell us for each pair of loci on the DNA, how many reads do we have? If we have more reads, that means these 2 loci are near each other because a lot of hybrid product is formed and you have a high read count. But if you pick 2 specific loci and you don't get many reads, so this means that the loci are probably far apart in 3 dimensions. You don't get a lot of cross-link product from those 2 locations. So now when you look at this heat map, first thing you notice that the diagonal is very bright because the diagonals are DNA that are near each other on the genome sequence. And if you are near each other on the genome sequence, you are also guaranteed to be near each other in 3D because the chromosome is just one polymer. If you are nucleotides that are within 10 base pair of each other, you are going to be near each other in 3D because you are only 9 nucleotides apart. But if you are looking at nucleotide at base pair 1 and base pair 10,000, it's 10,000 nucleotides apart. So depending on how the chromosome fold, these 2 loci can be far apart or it can be near each other, depending on how the chromosome fold onto itself. So when we analyze the chromatin conformation capture data, the first decision that we need to make is how high of a resolution can be analyzed. So in this sense, resolution is the size of one bin of genomic region. So imagine that when you map reads to genomic position, and you want to say that 2 genomic positions are near or far away from each other, because the reads span multiple nucleotides, so you can think of a unit of interaction as being multiple nucleotides. So usually people will use 1,000 base, 10,000 base, or up to 1 megabase. If you increase the size, right, you go up to 1 megabase. This means that you count all the reads that map to 1 megabase region at just one interaction. You're going to get a very low resolution result, right, because your DNA is going to be cut into few pieces. But if your data allow you to look at 1 kilobase as a unit of interaction, you're going to have a very high resolution picture of the data, because now your 3 billion base pair is cut into 1,000 base pair at a time. You're going to get a lot of region, a lot of bits, and you can see a larger image, a more detailed image. But the problem is, if you increase your resolution, you get a better image, you get more pixels. If you imagine as 1 bin of DNA, 1 bin of the genome as 1 pixel in this image, as you increase your resolution, your number of pixels is going into the quadratic. It's going to the square of the number of bits. So this means that you need high sequencing depth to fill in the data. So if we imagine that to study the chromosome structure, like to study the interaction between two loci, it's like you're filling the numbers, you're filling the data into this n by n matrix. So if your matrix gets larger, you have more and more pixels, you're going to need more read count to fill in the numbers inside. So this is part of the reason why you don't see as many chromatin conformation capture analysis as like DNA sequence, as like regular DNA sequencing, like exome sequencing or RNA sequencing. Because for exome sequencing and RNA sequencing, if your genome is larger, the number of reads you need scale linear with the chromosomes, with the DNA size. If your genome is twice as large, you need twice as much sequencing depth. But for chromatin conformation capture, if your genome is twice as large, you need at least 4 times as much sequencing depth. So when you scale to like mouse or human, which have very large genome, the cost of sequencing can be very prohibitive. Because if you do low throughput sequencing, your data is going to look like the example on the left here, which has many spots where you have maybe 5 read count, 2 read count, 3 read counts. It's not good enough for you to understand the overall 3D structure of the chromosome. So when we have this kind of problem, whether the question of do we have enough read, that reminds us of the situation that we had with metagenomic sequencing. We want to know for metagenomic sequencing, we did a visualization called rarefaction, where we want to know if 1 million reads is good enough to study the diversity of bacteria or microorganisms in the sample. And doing rarefaction, we try to reduce the read count, and then we see how many taxa do we still identify on the y-axis. We can do something similar for chromatin conformation capture. If we want to know if we have enough read count to analyze the chromosome structure at maybe 10 kilobase resolution, or maybe 20 kilobase resolution, or maybe 100 kilobase resolution. We want to know which resolution is okay for our data. So what we can do is we can vary the resolution. So we can bin the reads into 10 kilobase or 20 kilobase or 30 kilobase or 40 kilobase. So the resolution we can try at multiple resolution. And what we want to know is, okay, let's say we have 1 million reads. We're going to split 1 million reads into 2 halves, 500,000 each. And for each 500,000, at a certain resolution, we're going to analyze the data. So we're going to do 2 analysis, right? 2 replicate, where we divide the data into half. So it's like bootstrapping, but we specifically choose half. So we're going to analyze both halves of the data, and then we will compare this image that we obtained from one half of the data to the same image obtained from the other half of the data. If your data is good enough, if you have high enough sequencing depth, you're going to see similar pictures from two halves of your data. But if you don't have enough sequencing depth, like your data is going to be very noisy, you will not get the same result from your two halves. So the idea for this step is to split your data into two halves, analyze it, and then compare the variation that you get between the two. And for example, this graph on the y-axis shows the reproducibility between the result that you get from the two halves of your data. Higher the better. So the resolution, like if we go to high resolution, like 10 kilobase, we found that the reproducibility is like 0.5 on the orange graph, like 0.55 on the red graph, and then 0.75 on the blue graph. These are different algorithms, different analysis. And as we reduce the resolution from 10 kilobase to 20 to 30, the bins get larger. We don't get as high resolution. You see the reproducibility score becoming better and better because now you have enough data to analyze at this resolution. And what we want to identify is the resolution where you start to see the graph plateau. This means that even if you lose more resolution, you don't get more reproducibility. So your data is already good enough at about 40 kilobase. If you reduce to 50, based on the red and the blue algorithms, you don't gain anything. You lose resolution, but you don't gain much reproducibility. So this will allow you to select the appropriate resolution for your data. And there are many tools that will do this for you. And the only thing you need to do is to split your data into two halves. And these tools use different analysis to visualize the 3D structure, the 3D interaction. And your two halves of the data, the result will be compared, for example, by correlation or by the difference. And this tool will give you back how reproducible are the colors or other maps that you get from the two halves of your data. So these are just utilizing different techniques to calculate that. But the principle is the same. You divide the data, you look at the reproducibility. So this is the QC part of chromatic conformation capture data. In terms of data processing, it's almost identical to the pipeline that we already learned from variant calling from RNA sequencing. Because at the end, it's just pair-end sequencing. Right? And we want to know which part of the genome that the forward read map to, which part of the genome that the reverse read map to. So there's almost no spatial algorithm inside. The only thing that's done differently is because of the wet lab protocol for the chromatic conformation capture, where we induce cross-link and then cut with restriction enzyme, enrich with biotin, and then generate the hybrid fragment, and then do the pair sequencing. So during this process, there are going to be many unwanted products that are generated based on the pipeline. You might get a protein, you might get the hybrid fragment that are ligated the wrong way, or you might have a fragment that ligated in circle. So the only thing that are added to the bioinformatics pipeline to analyze sequencing data from chromatic conformation capture is how to distinguish between these products. So for example, if you can only map one end of the read, like we always drop them because we don't know where in the genome that the second part can map to. And the orientation can also tell us whether we are getting a circular product or a linear product. So the algorithm just check to make sure that the restriction enzyme that we use is near the sequencing site, because we know that the fragment, the hybrid fragment that we generate, both ends must correspond to the restriction enzyme cut site. So we kind of have some expectation of what product we want to analyze. Another additional problem is the sequencing bias. We learned from ChIP-seq lecture that different regions on the genomes have different amplification bias, different sequencing bias. So some region of the genome you will get more reads just by chance because it amplifies better than the other region, you get more reads. The same thing happens with chromatin conformation capture. Imagine that you have two regions of the genome that are amplified better than the other. So you need to produce a lot more interaction between these chromosome or genome regions that are easy to amplify. So people found that if you don't do anything to your data, you don't normalize anything, you're going to see this kind of a checkerboard pattern where some chromosome regions have very high reads. And when two chromosome regions both have very high read count, you could get even more read count from that involve these two segments. And people found that we can apply a simple technique where we imagine that the bias that we see in the interaction between loci A and B might be proportional to the bias at each loci, at each locus, multiplied together. Let's say you have like twice the chance of getting read from locus A and 3 times the chance to get the read from locus B. Together you expect to get 6 times more likely to get the read pair read, one coming from A, one coming from B. So people found that we can model this bias as a simple product between the bias for individual DNA locus. And after correction, we will see less kind of checkerboard pattern like the off diagonal here. The signal will be lower so that the two signals are usually inside the chromosome. So each of these boxes are individual chromosomes. We get high interaction inside the chromosome because each chromosome is a single molecule. Different chromosomes are different molecules. So the chance that they will be near each other in 3D is less than two different DNA on the same chromosome. And the chromosome coverage, like if we do the normalization properly, instead of getting some genomic region being highly sequenced, some being few, like we should get a roughly smooth genome coverage in entire data. So by getting to see like the interaction and the structure of the DNA of the genome of the chromosome like in this form also has other benefits. So for example, if you are studying a new organism and you perform the genome sequencing and you want to assemble the genome, sometimes you can make this kind of mistake. Because when you sequence the genome and you want to assemble the read, like when we learn read assembly, it's based on the similarity in the sequencing, in the sequence. But imagine that if your genome has two regions with similar sequence, there's a chance that when you assemble the read together, you put a segment from one chromosome to a different segment on a different chromosome because that sequence looks similar. When you look at the similarity in the DNA sequence, you don't know that you are merging two chromosomes together. But because chromatin conformation capture focus on the 3D structure, so when you map your sequencing data from chromatin conformation capture to the genome, if the genome is incorrect, this situation will occur. So in this case, doing genome assembly, we thought that this is one chromosome, but in fact, we are merging two different chromosomes together, which might share some repeat sequence or some similar sequence in the middle. But once we visualize the data based on chromatin conformation, we found that there is this region. Like you can see that the yellow signal like that signify the DNA that are next to each other and near each other in 3D. Like it completely disappears at this point. And also here, you don't see the continuation of the pattern. So this tells you that you have a misassembly. You have two chromosomes connected to each other at this position. So we can use this information to reanalyze and to reassemble your DNA. For another example, doing genome assembly, especially for unknown, for new organisms, you are going to generate many small contigs. So small contigs, these are DNA sequence that you cannot connect to anything else because you might not have enough sequencing depth, so you don't have the coverage of the genome. So you're going to generate these many small contigs. But using chromatin conformation data, you can see that there are some off-diagonal like yellow signals. So these off-diagonal yellow signals means that this chromosome that you already have, this contig, and this contig are near each other in 3D. So off-diagonal chromatin conformation signals in this graph, this plot, tells you that some of these contigs are actually on the same chromosome molecules. And using this information, some people develop bioinformatic tools to guide this process. You can reconstruct all these small contigs into a large chromosome. So chromatin conformation capture can be really useful if you are studying non-model organisms because it helps you organize the genome data when you don't have any other events. Also, when you study metagenomics, one of the problems in metagenomics is you're getting DNA from a whole bunch of bacteria. And if you want to reconstruct like the strain of the bacteria, you can have problem where you connect, if, for example, if there are 3 strains of E. coli in your sample, 3 strains of E. coli are going to have very similar DNA sequence. So when you assemble the DNA, you might making an error, like you might mix read from E. coli strain A to read from E. coli from strain B because they look so similar based on the sequence, you have no idea. But using chromatin conformation capture, only DNA from the same nucleus should be able to interact with each other. So by using chromatin conformation capture on metagenomics data, you can see that DNA from bacillus only interact with DNA from bacillus. DNA from E. coli only interact with DNA from E. coli. And for small plasmid, you can even tell that this plasmid is probably from E. coli because you see 3D proximity between plasmid F with E. coli genomes, but not with Vibrio and not with bacillus. So chromatin conformation capture can also tell you the relationship between plasmid or other mobile genetic element to their host as well. But imagine that how much sequencing that you need to perform, like to cover multiple bacteria, multiple plasmid to see this whole matrix. So the limit is the resource that you have. But in conceptually, you can map plasmid to the host by using chromatin conformation capture experiment. Okay, so from this point, we will talk about once we have these chromatin conformation capture data, how do we interpret the data to understand the 3D structure of the chromosome? So there are 2 primary structural features of the chromosomes that I think you should be familiar with. The first one is called the AB compartment. We'll talk about what it is. And the other one we already mentioned, topologically associating domain, or TAD. So let's start with the AB compartment. So what people found when they start to acquire chromatin conformation capture is that the interaction between DNA or the genomes are usually segregated into 2 groups, A and B. They just call A and B because of a lack of better name. So they found that regions of the genomes can usually be classified into 2 groups, A or B. And the thing is, DNA in the A region will interact with DNA in the A region. And DNA in the B region will also interact with DNA in the B region. So somehow, they found that the DNA, the chromosomes segregate itself into two classes that are only near the DNA from its class in 3D. And people later found that this actually corresponds to euchromatin and heterochromatin. Because we mentioned earlier that the heterochromatin, chromatin that will be inactive, these are recruited to the nuclear lamina. So they will be near each other in the near the nuclear lamina. And the euchromatin will be floating around in the middle of the nucleus. So that's why when you have chromatin conformation capture, usually the chromosomes will segregate into two groups depending on the chromatin and heterochromatin state of the cells that you are analyzing. And people can discover this with principal component analysis. I realize that we haven't talked about in detail about what this technique does, but the idea is you can analyze your chromatin conformation capture data through principal component analysis, and the first 2 or 3 components that you are getting out, one of them usually corresponds to euchromatin, one of them usually corresponds to heterochromatin. So in this study, they show that if you analyze the data, identify the A and B compartment, you can confirm that they are indeed euchromatin and heterochromatin by comparing to the histone methylation profile. So H3K4 methylation 2 correspond to one either A or B compartment. So this means that the overall structure of the genome is organized broadly into heterochromatin and euchromatin on the big scale. And I mentioned that when we zoom in, like from the chromosomes, we saw the compartment, euchromatin and heterochromatin. Once we zoom further onto the chromosome, we're going to see the topologically associating domain, or TAD. So TAD can range from tens of kilobase to megabase. So the size can vary a lot from organism to organisms and from DNA region to DNA region. So why are we interested in TAD? People found that when the genomes want to activate or deactivate the DNA to turn it into heterochromatin or euchromatin, we know the mechanism. It has to be recruited to the nuclear lamina. But what people later found is that this recruitment occurs not on individual DNA position, but on the TAD as a unit. So when a TAD changes from euchromatin to heterochromatin, it changes together as a block, not just a piece at a time. It changes together as a block. And furthermore, people found that during evolution, if you compare DNA from, from mouse, from dog, from rabbit, people found that if you perform chromatin conformation capture on these related animals, you can identify TAD that are conserved across animals as well. So you can see that there are 3 boxes of dense interaction that you can see in all 3 animals. There might some animal may have shorter or longer chromosomes, but the organization of the TAD into 3 blocks are conserved across evolution. So people found that the organization of the DNA is also encoded inside the DNA sequence. So there's some genetic element that tells the cells that this is where one TAD should be, this is where another TAD should be. And that's why it's able to conserve across organisms. Because the only thing that you can transmit across evolution is the sequence. You don't transmit epigenetics, you don't transmit other steps, you transmit the DNA sequence. And later we will answer what genetic elements are responsible for defining the TAD. So in terms of bioinformatics, how do we look at this image and tell that there are 3 TAD? We need to have some kind of score, some kind of algorithms to detect the TAD. And in this case, the intuition is simple. When we look at this chromatin conformation capture profile, in your mind, you can already make out some of these TAD because they are bright red on the diagonal and the off diagonal are lighter. So you see the boxes. It has to be dark in the middle and then has to be light on the outside. So a simple score is that if you focus on this yellow box and you think that the middle of the box here, where my mouse is a boundary between two adjacent TAD. If it's a boundary between two adjacent TAD, that conceptually we expect to see this kind of pattern. The A region, which is the diagonal, should be very dark. The D region, which is also the diagonal, should be very dark. But the B and C, which is off diagonal, should be very light. So if you count the number of reads inside A, add to D, and then subtract it by B and C, the score should be high if you are at the boundary between two TAD, because that's where you have dark, dark, bright, bright. But if you move your boundary to the middle of the box, you're not going to get as clear dark, dark, bright, bright pattern. So your score will be low. So as you move your boundary position of the TAD boundary along the genome, you're going to get high peak at where the true boundary is, maybe some for positive with some score, and then very high peak where the boundary is, and then high peak where boundary is. So we can turn the algorithm of detecting TAD boundary into a peak detection algorithm on this kind of signal. So the idea is we take the peak location as our candidate for the TAD boundary, and as a background to perform statistics, we already have these as a candidate. So to test it, whether these are significant, we compare to the score from other region, randomly selected region, maybe using just U-test to show that the score that we get from these peaks are significantly higher than score from everywhere else. And we suggest that these are location of the TAD. Very simple. This is just one of the many algorithms that you can use to identify the TAD. In fact, there are so many algorithms that no one can tell you which one is the best. So in this benchmark study, which is very nice, they try to utilize every existing TAD detection algorithms out there. And what they found is that the quality are quite similar. It's not like one algorithm is good, one algorithm is bad. But what happens is they make different decisions. Some algorithms make the same decision. They might have the same assumption, they may have similar algorithms. But some other algorithms, they are also performing equally well, but they produce different locations of the TAD. And you might wonder why, how can we have equally good prediction, but different prediction? So the thing is, the location of TAD in real life, in real cells, are actually not that clear cut. So in this example, one algorithm might define this TAD. But in your eye, you might think that, well, I might break this TAD down into two TAD instead. It looks like you have one TAD here, another TAD here. So the thing is, in cells, the mechanism that form the TAD is not always perfect. Sometimes the TAD can be here, sometimes it can be here, sometimes it can change a little bit. So when we analyze chromatin conformation capture, remember that this is not single cell data. So the average, the interaction that we are seeing here is being averaged across thousands of cells. So some cells might have this big TAD, some cells might have the smaller TAD. So the problem that every algorithm faces is that they are dealing with a mixture of TAD possibilities that are aggregated over many cells. So some algorithms will focus on larger TAD, some algorithms will focus on smaller TAD. So that's why we have different algorithms producing different predictions, but they are all looking equally good because they make different trade-offs. Okay, so that's the organization of the DNA 3D structure. So people from the physics side, they also want to know if can we model the 3D structure, the actual 3D structure, the position of each nucleotide computational. The AB compartment and TAD are like conceptual idea, like theoretical idea that you can apply to the heat map to understand the regulation unit of the DNA. But you don't get to see the 3D structure. This module is about 3D structure, so we need to think about, can we see like chromosomes or DNA in 3D? And they are indeed possible, but there are many limitations. So let's start from like early on, like 10 years ago, 15 years ago, people try to understand, is DNA, is chromosome, like, do they fold randomly or do they fold as with some organization? So we already know from our earlier discussion that if the DNA fold randomly, then you will get a lot of entanglement like this, and you will never get cell division in time because you need to unwind all the DNA from each other. So early on, people from physics already have some simple model that says that maybe the DNA has a fractal structure. Then you zoom in and then you see the same pattern over and over. Or maybe the DNA kind of fold in half at a time. The nucleotide that are next to each other fold onto itself, and you keep repeating this process. These simple mechanisms is already good enough to ensure that the chromosomes are separated from each other and form a nice region inside the nucleus. So even from 15 years ago, we have kind of simple idea that can explain how chromosomes can be so organized. But we want more details. We want to know how flexible can they be? Not just an average structure, but we want to model the actual DNA in one nucleus. So people use a lot of data from yeast because it's easy to study, and yeast only have 3 chromosomes, so it's not as complex as human. You have 23 polymers inside. So for yeast, it's only 3. So the early models model the nucleus as a sphere, and then they fix the position of the centromere because we know that centromere aggregate at certain location, and they let the rest of the chromosomes be flexible, so it can fold anywhere inside the nucleus. So early models can already confirm that once you fix some constraint, like you fix the size of the nucleus, you fix the positioning of the centromere, and you know the length of the chromosomes, you can already reproduce some patterns that occur in real cells. You can see the aggregation of telomere, even though there is no constraint on where the telomere should be. And by restricting the structure a little bit, you can already reproduce observation like this, that the centromere should be aggregated near each other. But it still doesn't explain organization like TAD. How do you control the cells to form certain domain on the DNA at specific location? Because these are, at the end, these are just random polymers. You are not guaranteed to see the same folding of the TAD. So later on, people thought that, okay, we think we identified some protein that might be responsible for organizing the DNA structure. And this protein can bind to DNA. So early models, they think that, okay, maybe if we think about the DNA as a string, and we think of protein as a binder. So if the protein, it can move in 3D, once it's found the binding site, if you bind to it, and once it also together, the protein and the binding site, together, when it meet another binding site, it can also bind to the second binding site. So basically the protein recognizes 2 regions on the DNA. And this causes DNA to clump together at certain locations. And those locations, the boundary of the TAD, must contain some binding site of some protein. But back then, like, people still had no idea like how many protein families are responsible for the folding. Because if all TAD, like if all domains require the same protein, how can you have such organized structure? Like, because sometimes you can have a protein bind to this boundary and then another boundary far away. So you should be able to form every combination of TAD. But instead, you don't see all combinations. Some TAD are very stable. So that must be something that specifically say that this boundary and this boundary must be next to each other to form this domain and not to other boundaries. So early on, people thought that maybe there are multiple protein families. So they say that one protein binds to one motif, another protein binds to a different motif. And that way you can have separate TAD that doesn't interfere with each other. But imagine, in the cells, if you have 10,000 TAD, do you need to have a family of protein with 10,000 binding sites? That's not possible. But still, 10 years ago, that's the best model that people had. So for example, like if you want to follow this kind of idea, you want to allow protein to bind to certain position so that it can regulate the structure of the DNA. You can run this kind of simulation. So one of the difficulty, like some of you might have seen a structure model for protein before, and you might wonder why don't we see structure model for the chromosome. One of the problem is the number of atoms that you need to consider. Usually proteins, like you have maybe 1,000 residues, like 1,000 amino acids, that's already large protein. But if you want to model the structure of the chromosomes, it's a megabase. So you have 1 million nucleotides. You cannot run a simulation with 1 million units very easily. So usually what people do is they will do what's called a coarse modeling. So instead of treating one nucleotide as a unit of the simulation, we will take about 150 nucleotides, 150 base pairs. And the reason is this is about the length of the DNA that are bound to one nucleosome. So if we think of DNA as being organized around the nucleosome, we can imagine nucleosomes connect to each other, being our building block for the DNA. So instead of 1 million nucleotide, we only need 7,000 units of nucleosome. But still, that's still already much larger than protein. But with simulation, you can show something like this. This is a simulation that I ran myself several years ago. So with protein in the dynamics, you can see that the protein induces the formation of two domains, two TAD. Without proteins, you don't get anything. It's just DNA folding randomly. And some of you might be wondering, like, how do we get started modeling something that has as many as 7,000 units? So people sometimes use what's called a self-avoiding random walk. So I think we heard the term random walk before, which is like a particle that can move anywhere in any direction with equal probability. We can also initialize, like generate a random guess of the structure for the DNA by using random walk. So we start from nucleotide number 1, or in this case, nucleosome number 1. We can pick the direction in 3 dimensions to go at random, either plus x minus x, plus y minus y, plus c minus c. We can go the initial structure randomly. And people call this self-avoiding random walk because we know that two nucleosomes cannot occupy the same space. They will collide with each other. So self-avoiding random walk, as the name implies, as you generate the random position, you try to avoid itself. You don't want to run into the original position. So you avoid the existing nucleosome. And once you can generate this kind of initial position, you can relax it into this kind of angled formulation. And this is because people have studied the structure of nucleosome to nucleosome. And they found that the angle between two nucleosomes, I don't remember exactly, but it's about 100 degrees. So you can rewrite the structure so that instead of a right angle, you can make them form about 100 degree angle from each other. And you get a very realistic random structure of the chromosomes. But then you need to update this structure to see if there are proteins, if it moves, like what will happen. So we have to think about the law of physics. Which law of physics can apply to the structure of the chromosome. So next class, when we talk about protein, there will be more details about the force, atomic force that acts on the protein, because different side chain have different properties. But for nucleotide, when we model nucleotide, in this case we model the nucleosome, we don't assume it to have any charge. So there is no specific electrostatic or bonding between them. But instead, we're going to consider that the molecule can flow. Imagine that these are molecules inside the nucleus. There are liquids in there. So the molecule can diffuse. Entropic force is a diffusion force. It can move each atom, each unit of the nucleosome in random direction. And because nucleosomes are connected along the DNA strand, there have to be tension force, similar to when you pull two sides of the rope, like you have a tension in between. And when two nucleosomes are too near, too close to each other, there have to be some repulsion. This is a basic force in physics. And in this case, there is a synthetic force, the one in green, called attraction force. And this force, which acts between not adjacent, but two nucleosomes that are one unit apart. This is a synthetic force that is used to control this angle delta. I mentioned earlier that people know from experiment that this angle delta, this angle theta between nucleosomes is about 100 degrees. So in order to control the structure to have about 100 degrees at this angle, people have to add a synthetic force, attraction force, to control this distribution. But otherwise, entropic force is diffusion of DNA in liquid solution. Tension is the force along the DNA strand, and repulsion is when two molecules of DNA comes near each other. So these are basic forces that we know the formula for. So we can kind of do the simulation by adding this. We'll talk more about how to do these kind of force-based simulation in the next class. Okay. So these are like the model from 10 years ago where people thought that there are proteins that help with forming the TAD, but it still doesn't perfectly explain the TAD structure. So a breakthrough comes under this model called loop extrusion model. So this idea originated when people found that the protein that we thought can bind to the DNA to help the folding, it doesn't just bind to the DNA. It acts like a motor protein. It can move the DNA when bind. So this idea is called loop extrusion because this protein moves the DNA so that DNA becomes a loop. So it extrudes the DNA into a loop. So they call this a loop extrusion model. So these proteins were identified the protein itself was identified many years ago, but the activity of the protein, the fact that it can move, it can walk on the DNA, was confirmed experimentally just 8 years ago in this experiment in bacteria, where they found that condensin in bacteria can move the DNA alongside the chromosomes and form larger and larger loops via this kind of time series chromatin conformation capture. So now people realize that it's not just binding to specific loci. Once the protein binds, it can move the DNA. So 8 years ago, this was published simultaneously in Science and Nature. So they accept this loop extrusion model as the explanation, the best explanation that we have for the formation of TAD. So what happened is on the TAD, on the DNA, there is this CTCF binding site, the one that we mentioned that are frequently mutated in cancer. So this CTCF binding site is bound by a protein called CTCF. And this protein CTCF is very special. It has a direction. So when CTCF is bound to the DNA and protein that can move the DNA, when the protein that can move the DNA run into CTCF, if it's run into the right direction, it will be stopped. So CTCF will stop the movement of the protein, in this case. So they are called condensate and cohesive. So the CTCF will stop the movement of condensate and cohesive. And this is why you get a stable boundary for the TAD, because the CTCF basically says that you cannot move further than this position. So inside our DNA, human DNA and mouse DNA, we have a whole bunch of CTCF binding sites dotted on the chromosomes. And these are the positions that define the boundary for our TAD. But the funny thing is, not every species has CTCF, which we will see in later slides. In plants, in some other organisms, we're still not sure how it's organized, or whether the DNA is organized at all. But at least in the case of mammal, in the case of human and mouse, we have the structure for the CTCF. It's a protein in green that can recognize the binding site on the DNA. And this protein can block the movement of cohesive and condensate, which are the two proteins that can walk or move the roots on the DNA. So this allows the genome to organize the location of the TAD based on the positioning of the CTCF binding. So this occurs in human in mouse, but in plants like Arabidopsis, which has no CTCF, you don't see the same structure. You don't see this kind of a diagonal box-like pattern in this plant. So we're still not sure in other organisms what mechanisms are responsible for regulating the gene expression and the organization of the chromosome. Okay. So the last thing I want to mention is the cell-to-cell variation of the chromatin structure. I think since we already learned about sickle cell technology, I think we all have the expectation that nothing is constant across cells. So in this study, so they perform chromatin conformation capture on individual cells from mouse. So it's like a single cell chromatin conformation capture. And what they found is not too surprising, because different cells, if they are from the same cell type, they should be in the same state. So in this case, even though the different cells give us different chromatin conformation pattern, if you look at the heat map here, they don't look the same. But if we ask what are the organization of the AB compartment, euchromatin, heterochromatin, what are the location of highly expressed gene and deactivated genes, and the position of the chromosomes, even though the pattern might not be the same, they are still organized the same way. There are still A and B compartment, and one of the compartments is associated with high expression, the euchromatin. And the chromosomes are still organized in specific region of the genome. If you compare the 3D models from two different cells, you see that the 3D structure are nowhere the same, but you still have two halves of the chromosomes being active and inactive, separate from each other. So that's why when we analyze bulk chromatin conformation capture data and we identify TAD and identify AB compartment, the data can be noisy because we are seeing slightly different organization across cells onto the same heat map. Okay. So just to summarize, what we covered today is our first lecture on structure, and we learn about the impact of how 3D organization of chromosomes that we used to think of as just coding the gene. But in fact, the 3D structure has many functions. It's responsible for DNA packaging into small for the cell division. It's responsible for bringing genes that should be regulated into the right location of the nucleus. And it has a very dynamic change during development and cell division. So it's a different layer of omics that we can think about on top of mutation, transcription, epigenetics that we are used to. We learn about chromatin conformation capture assays that can allow us to study the 3D information, but it can be costly to perform. And we learn about the explanation for how we identify topologically associated domain, the loop extrusion model. And we see the cell-to-cell variation. That still preserve like the state, like the U chromatin, the chromatin, but the actual 3D structure can be different across cells. Okay, so that's everything I have for today. See you next class.